So I think we talked here last time, and we talked through polyadenylation, we talked through the capping process at the 5' end, and then there's one other thing that has to happen. In eukaryotic cells, once the gene is transcribed, you get a really pretty long pre-messenger RNA. It's a messenger RNA that's not set up to be used for transcription. Normally, if you tried to transcribe it, you would get defective proteins. The proteins wouldn't be functional. And the reason for that is on this next slide. You get this protein that's being produced, and messenger RNA that's being produced. And in the middle of the messenger RNA, there are going to be sequences that aren't normally used. And they've indicated those sequences here by this kind of coloration right here. And so we see that this pre-RNA has already been capped. It's already had the poly-A tail added to it on the 3' end. Now one other thing has to happen. And that other thing that has to happen is that part of the RNA, where I've indicated with those arrows, those parts of the RNA have to be spliced out of the messenger RNA. They have to be removed from the messenger RNA. Somebody in yesterday's class asked the question, they got a little bit confused in this process, and they said, so wait, there are enzymes that come and they actually chop these things out of the DNA, and that's not right. The DNA is not chopped out. If the system, if it evolved, then that would make sense that those sequences, those are basically sequences down in the middle that aren't necessary, and so if you're an evolutionist, you got to ask the question, why are they there? Because it makes the genome much bigger, and it makes each one of those nucleotides, it requires a lot of energy. So why are they there? Why are they maintained there? We know that genes that are not essential or not necessary for some function tend to get lost. They tend to get deleted or mutated in the chromosome. So why are those introns there and maintained there in the DNA? And so the process, though, of removing those intron sequences is from the pre-messenger RNA. All right, I got us back up just a little bit because there's some vocabulary here that you might not have gotten someplace else, and I want to make sure you get it. There's two parts to this gene that we're looking at as far as the coding sequence go. Some of those are called exons. And an exon is a part of the coding sequence that's going to be used that actually codes for the amino acids. Okay? So when it's transcribed, the exons are the part of the messenger RNA that is going to code for those codons that you know about. And then the introns, I'll just circle one of those. Those are interrupting sequences. They're in the middle of the exons. They disrupt the exons, and so they got called introns. They break up the exons, and ultimately you see in this figure that those intron sequences are going to be removed from the messenger RNA before the messenger RNA can be used. Okay? And so this particular messenger RNA only has two introns that have to be removed. There's one here and there's one right here, and they're showing you this process. It's called splicing. The messenger RNA is literally cut, and then it's linked back together, and the enzyme seals the backbone back together so it makes a contiguous piece, and then part of that message is kicked out, and you can see that happening right down here as that intron is kicked out of the messenger RNA, and that's just a little bit of RNA that's floating around now, and there are lots of nucleases that can chew up RNA. They chew it back up into nucleotides. It can be used to synthesize the next messenger RNA. Those nucleotides can be used to synthesize the next messenger RNA that's going to be produced. Ultimately, then, we get a messenger RNA that is then ready to be translated. But if this is a eukaryotic cell, which it is because prokaryotes by definition don't do this process, where does that messenger RNA have to get to be translated? It's got to get out to the cytoplasm where the ribosomes live. So either the ER, the rough ER, or those my tongue's all tied today. Those ribosomes that are out in the cytosol, the cytocellular ribosomes or the free ribosomes, has to get out there. Okay, and that's the reason that we put that cap on, and that's the reason we put the poly-A tail on, right? We talked about that last time to protect it from the nuclease. So all of this process happens in the nucleus. All of this takes place in the nucleus before the messenger RNA starts to move its way to the ribosomes out in the cytoplasm. So let me just give you a scenario to make this real life. Down inside of your cells, there is a gene that codes for a DNA repair enzyme. And this enzyme is really complex. It's actually got 8 different polypeptides that have to come together to do the process. But one of those polypeptides is coded for on the chromosome, and it's coded for on a piece of DNA. And when it's transcribed, it makes a messenger RNA that is I'll just use round numbers, 21,000 base pairs in length. Okay, 21,000 nucleotides in length. Where's my eraser? Because it's messenger RNA now, right? It's not base pairs anymore. So 21K nucleotides in length. Okay? In the middle of that messenger RNA are also 20 introns. 20 introns. That gene that they just showed you in the previous picture looks a lot like a yeast gene because yeast have very few introns. Sometimes the genes will have 0, sometimes they'll have 1 intron, sometimes they'll have 2. 4 or 5 is the maximum that yeast have, and probably that's because they're simple cells. But this gene has 20 introns in the middle of it. So the messenger RNA is made. It's 21,000 base pairs or bases in length. And once it's processed, it gets chopped down to a message that's about 2.1 thousand. So in other words, 90% of the pre-messenger RNA is removed in that process of eliminating those introns. So this is a gene that I studied in the laboratory when I was at Southwestern Medical Center, and I sequenced a bunch of the genes from different defective families. It's not a good way to say it. From families that were defective in this process. And so we looked at what was going on, trying to figure out what was going on with their genes. Why couldn't they repair UV light? And many of them had just one amino acid that the change had occurred. You can't see what's going on, but it's driving me crazy up here. We'll just go there. So there was one amino acid change, a mutation change down at the 3' end of the gene that just made a simple substitution. It was one hydrophobic amino acid for another hydrophobic amino acid. Very conserved. You'd expect it wouldn't have much function in change of the final protein that was going to be produced. And so that was hard to explain, but some of the others actually hit some of the junction sequences where the intron splicing was. And when they spliced, they actually misspliced, and so they cut off parts of the gene. And so those splice sequences, what I'm trying to say is these splice sequences right here and right here are very, very important. Again, they're enzymes that have to come and recognize that sequence in the RNA and recognize that sequence in the RNA very precisely cut there, because think about what's going on. If it cuts one base or 2 bases on either side of where it should cut, it's going to result in a frame shift. It's going to result in the reading frame shifting out of frame, and then it's going to make a completely different protein once that splice happens, if it happens incorrectly. So those enzymes have to recognize very precise sequences here and here. They have to cut very cleanly there, cut very cleanly there, and bring the things back together. Or otherwise you get a defective RNA that's going to make a defective protein. And if you've got 21 of them in the middle of a gene, you've got to start asking the question, why would you do that? It makes 21 times where you could make a mistake in the splicing and make a defective protein. And so the answer to that question is because it gives a lot of levels of control. One thing is that splicing process takes time. And the more you have to do, the longer it takes for the messenger RNA to get out to where it needs to be. And so that gives a little bit of a level of control. But if you think about it from a different perspective, there's something that's very interesting. Remember what we said about bacterial messenger RNA last time? How many bacterial messenger RNAs are polycistronic? Do you guys remember what that means? What does polycistronic mean? I think I drew a picture for you last time. It's been a while. Yep. So many genes. And what does that mean when we're thinking about messenger RNA? I think I drew it right over there. Try to jog your memory, unless I drew it on here. That's right. So somebody in the other class asked about ORFs. Remember that term, ORFs, O-R-Fs? Yeah, you probably pronounce it ORF. Usually we don't pronounce the letters, we just say ORF, which stands for open reading frames. And that's the beginning of an ORF is a AUG, the end of an ORF is one of the 3 stop codons, and in between is a group of codons that can code for a protein. And so on a polycistronic message, we're going to have multiple ORFs on the same message. So when you want to kick on a gene or a process, if you make a long messenger RNA that has multiple open reading frames, you can start making all of those proteins at the same time. So let's say lactose utilization. We're going to talk about it in a bit. But lactose utilization requires 3 enzymes that aren't normally turned on. And so they're all coded for on the same messenger RNA. As soon as lactose is in the environment and there's no glucose, they'll kick on the transcription of that particular messenger RNA, and it'll make all 3 of those proteins real quick. Anyway, that's polycistronic. And then we said there's another term that describes the typical eukaryotic messenger RNA. What was that term? It's kind of the opposite. Monocistronic, good. Monocistronic means there's only one open reading frame on that message, except if you do something different. So I came back to this slide to show you this slide because it's easy to look at on here. So here we've got a gene that's got 2 introns. Think about this. If this particular cell decided sometimes to splice out both of those introns, it would make that protein that we just described from this messenger RNA right down here, right? But what if sometimes this cell doesn't splice out this one? And if there were exactly some multiple of 3 nucleotides in there, 33, 333, whatever number you can think of that's divisible by 3, if the nucleotides that are in that intron are codon links, what other numbers? 333 is an easy one, but any number that you can divide by 3, so 300, 210, anyone that you can think of that could be divided, that will make this part translate, and then it puts in some more nucleotides, and I'm just going to put those squiggly that aren't normally in the protein, and if you have that division by 3, it'll go back in and it'll make the protein look just the way the original gene would after that. And if this intron splices out, then the rest of the protein that's produced will be exactly like the original protein that you predicted to be produced. And so you can get lots of different alternative proteins out of one messenger RNA depending on how they splice. And if there aren't stop codons in the middle of those introns, or the number of codons are the right so that they're divisible by 3, you can get a functional protein that comes out. And so you can actually get multiple proteins that serve the cell with similar function because some of the domains on the protein are still the same if that process that I just described happened. Now there's something new, this part right here, that's stuck in the middle of that protein, some amino acids that aren't normally there, that presumably can have some other function. And so even though we say eukaryotic messenger RNAs are monocistronic, you can many times the cell gets different proteins out of them depending on how they splice out those introns. So it gives the cell some alternative function for the same messenger RNA and potentially allows that process to happen. They also, some of the processes just shutting down other normal processes. If you misfold or miscode the protein by leaving one of the introns in, I see your hand, I'll get you in a sec. If you leave the intron in and get something added, it may go bind to one component of a process, but then it'll block that process because now it doesn't work with the rest of the components that are involved in that process. Question was?
All right, I don't remember if you already answered this, but the introns,
Mm-hmm.
do they contain start and stop codons?
So typically, well, the answer is yes, they can. They many times do contain stop codons if you stay in the same frame, but if they don't, then you can get this scenario that I just described to you where you can get alternate proteins coming out. Yeah, so the start codon, usually the ribosome binds to that ribosomal binding site that we talked about last time. It binds there, that's close to the 5' end of the messenger RNA, and then the ribosome slides along the messenger RNA, and usually it starts translating at the first start codon, the first AUG that it bumps into. So there's going to be lots of other start codons because any AUG codes for methionine. If you go back to the genetic code, there's only one codon that codes for methionine. So there's going to be other AUGs in there because methionine is one of the 20 essential amino acids. So technically it's not a start codon at that point. We consider the start codons those AUGs that are right there by the ribosomal binding site.
That the introns, are they present there because on eukaryotics it's more compartmentalized in DNA, therefore it goes for more proteins that are specific for other functions rather than the one that is specific for that protein that is supposed to be made?
So let me make sure I understood your question. The first part was eukaryotic cells are more compartmentalized, so proteins need to go to different compartments of the cell and be functional there.
I mean, it's like, you know, if you look at DNA, right,
Uh-huh.
DNA is very specific to, you know, individual functions within the cell. So those introns, they have to be present there at the DNA level. And you know, if I'm getting this right, those introns are part of those individual messages that create, you know, the compartments within eukaryotic cells. So they have to be broken down before they actually become specific.
They definitely have to be spliced out before they can become functional, but my mind went to the opposite scenario, what you said, the introns have to be there. And that part's not exactly right, because the way that we found the defective genes, if you've got an individual that can't do a function, you've got to figure out some assay that you can figure out which gene is actually defective. And so the way we figured out which gene was defective was we had what are called cDNA copies of each one of the messages. And so you take the messenger RNA, you make a DNA copy, you throw it back into the cell, and then if it's transcribed and fixes the problem, then you know that that was probably the defective gene that was in the cell. And you can make libraries of these, and you throw them in until you find a cell that's actually able to do what it couldn't do before. You harvest that cell, and then you pull that DNA back out, and then you can look at the DNA and say, okay, what's there? What kind of new DNA? What did it get that made it functional now? And those cDNAs are made from these RNAs that are already spliced and already processed. So you harvest out the mature RNA, then you make a DNA copy, and then you throw that back into the cell. And so the introns don't have to be there, which is what I thought I heard the first part of the question. They don't have to be there. If this gene existed on the chromosome, and that's done all the time, without those introns, it'll still make a functional protein. Because the introns are there just doing something, and presumably they're doing something between these two steps right here, unless one of the introns doesn't get spliced out. And the enzymes that come in and splice are more selective for some of those splice junctions than they are for others, because there's slight variations. Some of them splice out faster than others, some of them splice out slower. So there's a little bit of predominantly regulation, but it also gives the cell the ability to make multiple proteins from the same gene, the same locus on the chromosome. All right, so switching gears a little bit, we're jumping back to bacteria now. And if you think about bacteria living in an environment, they've got to be able to respond to their environment. Microbial environments are complex. There's always changes in the environment. If you think about just an E. coli cell that lives down in the gut of a mammal, that mammal might be you. And if you think about him living in the gut of a mammal, sometimes you're hungry, you eat lots, Thanksgiving's coming up, I expect you'll do that. Other times, the week after Thanksgiving, you realize you put on too much weight, you decide to not eat so much, and so the bacteria in your gut then become hungry and they're not fed as well. So they really live in periods of feast and famine. Even if you think about a normal day, most of us eat 3 meals a day, and some of us snack in between. And so if we just eat 3 meals and don't snack because we're trying to lose that weight, there's actually not a lot of fresh nutrients that are going in to feed those bacteria that would be in the gut of a mammal. And so when the host is eating, the bacteria then can take up amino acids from the meal that's just been taken, other vitamins and other nutrients, and they don't have to synthesize those, so they actually shut down those synthetic pathways because they don't need to make them. They're being provided in the environment. There's no need to make those if they're out there in the environment, and then they can channel that energy into other processes like making DNA, making proteins, those kind of things. And so at that point, life is good for the bacteria, but then between meals, the nutrients become fewer, and at that point, the bacteria have to synthesize those amino acids. They have to synthesize those other vitamins and other things that they need, and so they have to activate those biosynthetic pathways, and those require energy. Energy is consumed when they're making those processes, and therefore growth is slowed down at those points. And then it gets even worse. The animal feels the urge and they go and defecate, and then the E. coli is released from the bowel of that animal, and now the E. coli is out in a completely different environment where nobody's feeding it. It's got to figure out how to break down cellulose or break down other things that are in the environment to get the energy that it needs. And again, it's got to shift, and it's got to kick on entirely different sets of genes to be able to do those processes. So that lecture slide is designed to make you feel sorry for bacteria, because they can't just walk to the cafeteria. They're really happy when you decide to eat them and give them a nice warm place to live and grow and have lots of nutrients. And if you never decide that you're too overweight, then they'll be really happy. They'll just love you for feeding them all the time. Some of them still end up being released from the body either way. This slide's to make you think about, yes?
That's not probably a very important question, but like the people who are anorexic, do their bacteria die if they don't?
Well, they're going to have less. And there's definitely different populations that are going to be there. I just saw a couple of different studies at this conference that are looking at that. Remember, there's this massive human gut microbiome project that's going on to figure out what kind of organisms are in people who have too much weight and people who have too little weight, and describe normal people. And so there's going to be different populations, but there'll definitely be less, because people who eat less, their intestinal tract tends to also decrease in diameter, and so there's going to be less bacteria that are there, and they can't maintain if they aren't being fed, so they're going to be eliminated from the body. So remember, on the Facebook groups, somebody found, maybe I found, the video of a little bacterium. It was a Diplococcus being chased around by a neutrophil. Remember that? And when you watch that, did you ever wonder, how does the bacteria know that the neutrophil is getting close? The neutrophil would get almost right up on it, and the bacteria would zip off in a different direction. And pretty soon the neutrophil would make a direct left turn if that's the direction the bacteria went. And that has to do with this topic. How do they sense their environment? It was almost like they had eyes on the outside of their cell, and they could figure out which direction they were going. And so there's two mechanisms, predominant mechanisms, that bacteria and actually your cells also use to figure out what's going on in their environment. One of them is called signal transduction. And signal transduction is a system that allows something that's happening on the outside of the cell, some protein, some chemical, something else, to be recognized on the outside of the cell. And in signal transduction, that chemical is not brought into the cell, but there are proteins that bind to it, and then they transmit the presence of that chemical, the fact that the chemical is present, to proteins that are on the inside of the cell. And so we'll talk about that in just a little bit, and we'll walk through, and then that allows the cells to react. So that's probably what was happening with the neutrophil as it was chasing that little bacterium around. And it's probably what was happening with the bacteria as well. The bacteria is shedding chemicals into the environment as it swims along. And the neutrophil has receptors on its surface, proteins on its surface, that are designed to bind to those chemicals. So some of you probably also asked the question, wait a minute, the neutrophil went just right past two bacteria that weren't even moving. They were just sitting there, and the neutrophil glided right past them on toward the one that was moving. That moving bacteria was shedding more chemical into the environment, and the chemical that's being shedded is probably LPS, maybe a little bit of peptidoglycan, and the neutrophil has receptors on the surface for both of those. And so it was picking up the presence of those chemicals and using them as chemoattractants to draw it toward those bacteria. And that's what happens inside of your body as well. And so that was signal transduction. The other process that bacteria can use, and we do this a little bit in our system as well, but it's more important for bacteria, is quorum sensing. So how many of you served on a committee before? Have you served on some kind of committee? Cool. And did your committee have rules on when you could make decisions? I'm on the faculty assembly. I'm the president this year, and we've got a policy where we can't make important decisions unless 50% of the faculty are present. So there's 163 full-time faculty right now. A couple Mondays ago we had a faculty assembly meeting, and there were only 76 faculty there, and so I was up at the podium hemming and hawing, hoping that 4 or 5 more faculty would walk in the door so we could make some decisions. We had some things that we needed to vote on, but there weren't enough faculty to allow us to vote and have it to be valid. And that number for us is 50%. For you it might have been something else. It's called quorum. And we just barely made quorum. Finally, one more person came into the room. We all celebrated and applauded. I said, please don't anybody leave. And then we quickly voted on a couple of things that we needed to vote on as a group. And that's called quorum. You guys have probably done it before too. You were up late studying and you wanted to go to Starbucks to get a coffee, but you really didn't want to go by yourself. So you called a friend and said, hey, let's go to Starbucks and get some coffee. I don't want to go by myself. Will you go with me? And that friend said, no, sorry, I got to wash my hair tonight. And so you called another friend, and she said, sure, let's go to Starbucks. But then the two of you said, well, I don't want to go to Starbucks, just the two of us. Let's call Susie or Sally and see if she'll go too. And you found a third person, and finally you went to Starbucks and you got that coffee that you needed. And that's kind of developing a quorum too. Well, bacteria do the same sort of thing. And you guys asked about costumes, so I'm going to tell you this part real quick. One of the costumes that was pretty cool is a friend from Texas Tech, and her lab decided to come as an organism that some of you might have accidentally looked at when you were doing exercise 3. On the Vibrio cholera tray, there are also slides for Vibrio fischeri. And Vibrio fischeri is an interesting organism. It tends to live out in salt water. There's a lot of it in the Gulf. And this guy is pretty interesting. It has the process of bioluminescence. It can take ATP and turn ATP into light. Like the firefly that we talked about before, it's got different enzymes that do it, but it only does it when there's a lot of the cells present. So the theory is that Vibrio fischeri lives in water, and when the population's getting too dense, they kick everybody into bioluminescence mode. That light in the middle of the ocean attracts a predator, which comes and tries to eat whatever's glowing in the ocean. It eats some of it, swims away, some of it comes out the intestinal tract in a new location, and it helps the organism to be able to be dispersed out into the ocean. Those new cells that got moved away now grow up in numbers, and when they get enough, they'll start making light. Somebody else will come and eat them and carry them away again. And so that process is called quorum sensing. And they don't need to move if there's not many bacteria there, and to make the light costs the cell energy. So they only make the light when there's a large number of the cells present. And that tells them, hey, if we keep growing at this rate, pretty soon we're going to run out of nutrients here. We've got to move some of us over to San Antonio or wherever it is down the road somewhere so that we can spread out the population a little bit. And so they all kick on the light all together. And that's done by this process called quorum sensing. And quorum sensing looks something, basically they sense the density of their population, and work with me. I think I just lost Wi-Fi. Okay. So here's a picture of the scenario of quorum sensing. If there are few bacteria present, like in this panel on the left, they're doing their thing. They're making products, and one of the products is a small molecule. It's a small polar hydrophobic molecule. Part of it's polar, part of it's hydrophobic. And you can see that molecule here. It's called the signaling molecule. They make this, but if there's only a few cells present, only a little bit of that material is produced. If there are lots of cells present, like in this right panel, then there's lots of that material produced, and the concentration goes up in the environment. That molecule is not the light-producing molecule, but it's what the cells are using to figure out how many of them there are. The other property of these molecules is that they diffuse easily through membranes. So if you have a high concentration on the outside of the cell, you also have a high concentration on the inside of the cell. And that high concentration on the inside of the cell acts to induce genes that aren't normally turned on, to kick on genes that aren't normally turned on, and those are the bioluminescence genes. They induce those bioluminescence genes. The whole population then decides, okay, we got to do it. There's that much chemical here. They kick on, they start glowing, and that's the process. So back to the costume party. One of the groups works on Vibrio fischeri. They all dressed in black, and they had these neon tube wrapped around their body. And so whenever they came together as a group, if they were sitting at the table, they would flip the switch, and all of their lights would kick on at the same time. But as soon as one of them walked away, they'd flip the switch again, and the lights would go back off again until they came back to their group. And so they won the costume prize because they had a functional microbial costume, and it was pretty cool. They were all dressed in black tights, and they had this kind of light chain wrapped around their entire body, including their arms, and they just had a little switch right here like Spider-Man so they could flick it on and off whenever they were coming together or going. It was pretty cute. And so, anyway, that's quorum sensing. That's how it works. Besides that process of bioluminescence, quorum sensing is also important for some other things biologically. Biofilm formation. It takes a ton of energy to make those polysaccharides and secrete them outside of the cell, or to make those polypeptides and secrete them out. And if you're just a few cells in an environment, you don't probably want to do that. You can't do that all by yourself. You need a community to be able to do that. So the production of biofilm is controlled by quorum sensing. When you get enough cells there, they start realizing, okay, we're enough, let's do it. And they start synthesizing all of those polysaccharides to pump outside of the cell. And that's very important